Good morning, everybody. On behalf of Hortgro and Standard Bank, I would like to welcome you to this Symposium webinar series. First of all, I want to acknowledge Standard Bank for their sponsorship, making these occasions possible. It became evident last year already that a physical symposium wouldn't be possible, but Hortgro decided we wanted to maintain a presence in the normal symposium week. Next year, we endeavour to have our symposium again, and for good measure, also the Hortgro Showcase. So those two events will hopefully take place again from next year on as physical events. But in the meantime, we're going to have the webinar series, interesting topics, and a lot of interest judged on the number of registrations. So back to the webinar series. This is the first of three. It's focusing on chill models, and if you've looked at the forecast for the next couple of days, the next week or two, it becomes evident why we want to talk about winter chill and chill units. I'm going to pass on without further ado to Dr. Esme Lowe. She's the chair of the Horticultural Science Department at Stellenbosch University. It's a department with a long history of conducting research, very applied stuff, and more in detail work, more fundamental work on dormancy, starting from the times of Prof. Strydom, Prof. Jacobs, Nigel, and now Esme. Esme will be the facilitator of the session. She'll introduce the presenters to you. Just one housekeeping rule. I would remind you to please keep your microphone switched off, to mute your microphones, and please keep your video also switched off. Questions can be posted in the chat box and will be put to the presenters at the end of the presentations. So now over to Esme and Nigel. I'll just vacate the chair and open it up for them.
Good morning, everyone. Thank you, Vian. So as Vian mentioned, I'm Esme. I'll be your host for this next 60 minutes or so as we try to unravel the complexities of dormancy and try to understand the challenges of farming and producing deciduous fruit with a warm winter climate. I'd like to introduce you to two guests. First of all, we've got Professor Eike Lödeling, who's joining us all the way from Germany. Morning, Eike. Eike is a professor in horticultural science at the University of Bonn from around 2018 onwards, and before that he spent time in Africa, in Nairobi in Kenya, where he was a climate change scientist and later a senior decision analyst at the World Agroforestry Center in Nairobi. In research circles, Eike is very well known. He's got more than 100 peer-reviewed published articles that are cited commonly and everywhere when one talks about dormancy and temperature modeling and the interaction between a plant and the environment. If you're looking for information on that, his name pops up all the time. He's also very involved with modeling of temperature data, and of late also trying to understand how tree dormancy fits into all of this. And then my second guest really needs very little introduction to the industry. It's Dr. Nigel Cook. Nigel started off as an academic in our department at University of Stellenbosch. But he's a very applied scientist. He, for the last 25 years, have had a horticultural production technology consultancy where he services local as well as international growers. He really understands the South African climate and what it takes to be farming deciduous fruit under our warm winter conditions. He's also a nurseryman and of late very interested in breeding of low-chill apples. So as far as expertise and knowledge and skill go, I think we are in very good company this morning. In terms of the agenda, I would like to just give you an idea of how the next 60 minutes will unfold, I will start off by doing an introduction on dormancy, sketch a bit of a background, just talk about the status quo of what we know about dormancy specifically in a plant, and that will flow then into a conversation that Aker will continue with chill models, and he will pass the baton on to Nigel, talking about how dormancy progression and chill models become problematic and why that is difficult and how that translates into the South African context. And then I will try and conclude and form a narrative from what we've discussed. And then most importantly, we want to open the floor for questions so that we can have a bit of interaction. And we welcome your questions. If you think about any questions, you can start typing them into the chat already, and we will make sure that those get out to the speakers. Without further ado, let's start with the introduction. If we introduce the topic of dormancy and we talk about where it comes from and why it exists, I think we need to take one step back. And I'm going to use an apple as an example because I think that's a deciduous fruit that is well known to all of us and arguably one of the more complex fruit types to work with. So apples are from Kazakhstan. That's where they originate from. The picture there on your left-hand side, that is a wild apple forest in Kazakhstan. The white that you see there are actual apple blossoms. So that's where the apple tree was born. Kazakhstan is a country right in the middle of the world. It's actually the world's largest land-embodied continent, meaning that there's no ocean or big water body around that can moderate temperatures. And that gives origin to what is known as a continental climate in that country, which basically means you've got big fluctuations, seasonal fluctuations in temperatures. In summer, you will often have temperatures very similar to the South African context, 30, 35, etc. degrees Celsius. And then in winter, on the other hand, you will have extremely low temperatures, easily below minus 20 degrees. So the apple is adapted to that, genetically adapted to these conditions. And I think we need to start our story over there to try and understand how, over time, the apple has been able to survive, especially those severe winter conditions. And this is where the conversation starts with dormancy. The apple tree, or any deciduous fruit for that matter, is adapted to inhibit its growth during dormancy. And just move on. And if we follow what happens in a cold winter area such as Kazakhstan, we will see that what happens during autumn as the colder temperatures set in, the plant senses this. Hardening off happens, cold hardening happens, the plant starts to form bud scales around its meristems to protect those vulnerable sites of growth from harsh temperatures, and the whole growth center shuts down during dormancy. That much we know. We know that there are physiological changes that happen in the buds to slow down growth and completely inhibit growth. And in a cold winter area, this happens relatively fast. The autumn is quite severe in compared to what we have in South Africa. Autumn is quite a sudden event. You have drops in temperatures, and that causes the cold hardening, as well as the preparation for the bud. The dormancy starts to set in, this inhibition to grow starts to form. It's a physiological block within the bud itself. And as we progress into dormancy and into the winter, this will increase. And once the bud has reached maximum dormancy, It actually requires low temperatures to break down that physiological block again. So chill is required to overcome dormancy, to actually overcome the physiological block inside the bud. And this is where we often talk about the chill requirement of a specific cultivar or species, because it needs a certain amount of cold, it needs that exposure to low temperatures for the bud to actually reverse the physiological block to such an extent that it can in spring, when warm temperatures set in, start to grow again. We know that chill requirement is fruit type and cultivar dependent, and that differs. And we also know that dormancy really is a survival mechanism, so that we don't have these meristems growing under unfavorable conditions. So how do we measure dormancy? How do we as humans try and quantify this dormancy that we see in plants? I want to give a quick rundown of how we do this, because I think this might be crucial for understanding some of the other slides that follow. So what we do as scientists, we go during this winter period, go out to the orchard and we collect 30 centimeter shoots, 1-year-old shoots from the trees. We put them in bundles back in the laboratory and we expose them to optimum growing conditions. So that would be enough water, enough light, as well as temperature, probably around 22 to 25 degrees. And then we monitor and we wait until these buds, terminal buds, start breaking and growing. And that amount of time that it takes for it to actually start growing is then an indication of how deep the tree is in dormancy at that specific point. So the longer it takes, the deeper it is in dormancy, the longer it takes for that physiological block to reverse and start growing again. Through this actual very simple method, it allows us to quantify what we see. So the graph that you see on your left-hand side actually indicates on the y-axis days to 50% bud break. So this is what I've been talking about, the time that we waited out. Every dot on that graph indicates a collection point of shoots, and then how long it took. And you can clearly see what there, we've got two graphs of Fuji from the Koa-Bokkerveld in 2 specific years. You can see that entrance into dormancy and how it exits again from dormancy. That's quite clear. Very similar to the previous slides that I showed you with that dormancy progression. We also know that this dormancy progression looks quite different when we are in a warm winter area. When the winter is not cold enough and we do not have enough chill units to fulfill the chilling requirement and to break down the physiological block, this picture looks a lot different. The entrance into dormancy takes much longer, and we all know the result of that. In the orchard, we will see delayed foliation, we will see very poor bud break, which will lead to mixed maturities, which is really not ideal, and from a management perspective in an orchard can be very challenging to manage. And yes, we can mitigate this using chemical rest breaking, but we also know that often that is a hit and miss, because we don't always understand the exact dormancy level that the tree is in when we apply these chemical rest breaking methods. And what we see on the right-hand side is the chill units accumulating. So what we do now is we look at the dormancy graph on the left-hand side, and we try and match it up with the amount of chill units or the amount of cold that that tree has experienced. And what we see there is a chill model. In this case, it's the Utah chill model of those exact 2 years, and you can see how chill is accumulated throughout that winter period. And there for a moment, I want to pause and just indicate, and just let us think about a chill model. Chill model is us humans or researchers' way of trying to gauge what the plant is experiencing. This chill that the plant needs, low temperatures, specific temperatures that the plant needs to break down the physiological blocks so that the inhibition will disappear and growth will happen. We are trying to measure and quantify that by using chill models. So it really is a tool that we use to try and analyze a plant's response to the environment. And over the years, there's been a couple of these models developed. And at this point, this is where I want to hand over the conversation to Professor Eike-Lederling to unravel and talk more to us about these chill models and how us humans perceive the way plants do things.
All right, thank you, Esme, and I'll be happy to comply with that request. So, I've been looking at these chill models for some time and have been trying to understand chill models for well, about 15 years now. I'm making some progress. I'm going to try and share some of that today. It's actually a pretty difficult topic. It's not quite as, it looks somewhat easy on the surface, but then once you dig in a bit deeper, it starts getting more and more complicated to some extent. We've already heard just now that, well, obviously trees go through the dormancy period in winter that we can sort of, let me show you something here. We can sort of conceptualize it as a process where at some point in the beginning here, in fall, in autumn, the trees enter dormancy, then they go through a phase we call endodormancy, where basically they can't easily grow even if we put them under warm conditions. At some point, then they transition into a different state, which is then the ecodormancy, and then basically they start waiting for warm weather and accumulating heat units, and then eventually they start breaking their buds. Now, this is how, in principle, we're pretty sure it works. And now I need to use the arrows. The main challenge here for modelers, of course, is that we don't really see much of a change during most of this period here. The buds look pretty much the same at the beginning of dormancy and at the end. There may be some slight changes in sort of the molecular level, but we can't really see much of that. And especially what we can't really see is that moment when they've actually chilled enough, when they fulfill their chilling requirements. This is basically something that we can't really directly observe. And so for modelers, it's pretty difficult to model stuff that we can't actually see. And we really need these rather complex experiments that Esme has just described in order to know when this chilling requirement has actually been fulfilled. And that's been a major problem really in developing models. Yeah, it's quite difficult to model the chill and also the heat accumulation process because we don't really see much of progress in the buds during that time. Now, of course, there are these chill models that I'm sure many of you are already using somewhat routinely. It's actually quite nice to be talking to a South African audience, which I'm very grateful for the opportunity to do this, because here in Germany, people aren't so interested in this topic because the winters are cold. We're not quite Kazakhstan here, but usually we don't really have a problem breaking chilling requirements. I know that this is a much bigger topic over where you are. And so it's nice to be able to talk to a real audience that cares for a change. Let me give you an example. Starting with the simplest chilling model, just to give you a feeling for how bad models can be to some extent, we have evidence from a bunch of studies that have measured these chilling requirements. Willie, would you mind muting your microphone again? Thank you. And for example, there's evidence from Turkey or from Iran that tells us that pistachios, just as an example, require about 1,000 chilling hours in order to fulfill their chilling requirement, allow viable production. A chilling hour is an hour with temperatures between 0 and 7.2 degrees, which looks really precise, but it's just a translation from 45 degrees Fahrenheit, which looks a lot less precise. Now, the big question, of course, that maybe some of you have experience with already is what would production look like if we only had kind of a small fraction of this? Would we really see major problems? For example, we could imagine a situation where we only have maybe 100 chilling hours or so. And what would happen then? What kind of production level would we see at that point? So that's the major question that these chilling models somehow have to help us answer. And I can tell you what this looks like. Here we have a pistachio orchard with about 100 chilling hours. So this is not in Turkey or Iran, where these 1,000 hours have been estimated. This is in Tunisia in North Africa. Does anyone see major problems with this tree? I mean, I'm pretty sure you're not, probably not, most of you aren't pistachio growers, but this is not a tree that's in trouble. Now, this is actually doing well, even with there's the same species and the same cultivar as in Iran and in Turkey, and there are no production problems here at all. But the chill levels are much lower. We only have about 100 to 200 chilling hours in this location here. So what does that mean? Yeah, so this is 10 to 20% of what's been estimated as the chilling requirement of these trees. Now, pistachios are doing all right, and this is evidence. I mean, scientists would try to figure out why this is happening. This can be evidence of course, some sort of intraspecific variation. Maybe these cultivars aren't quite the same, and they're doing something slightly different, maybe. Or they got used to these conditions, acquired thermal needs, so they know they grow in a warm place, and so they've reduced their chilling requirement. Could that be it? Or is it another one? Is it maybe an inadequacy of the chilling hours model? Maybe we're just using the wrong model for measuring this. And we can't really tell what the answer is. But I can tell you it is probably 3. Yeah? So it actually is an indication that this model is grossly inadequate for kind of quantifying chilling requirements across different locations. And this is really a problem with many models, and especially in this particular case, it's quite striking to look at sometimes where models come from. And this is important to understand if we want to apply them somewhere else. So what's the background of this chilling hours model? Well, there's one scientific reference we always use. It's a study by a guy called Weinberger from the US who published on this in 1950. But he didn't really do any experiments to develop this model at all. So there was no controlled process that this guy did. And he actually already showed evidence that this wasn't even working well back then. Still, it's been used for a long time. It's quite interesting to look at where this model really comes from, which is this reference in that paper that everybody always cites. And so basically, this is an unpublished paper, which basically means a talk that somebody gave in 1932. There's no record of this. We don't really know where this came from. We can look at who this was, but other than that, there's really no evidence of how this model actually came into being. And so here's the guy who gave that talk. It's a guy called Lee Milo Hutchins, who was a pretty renowned phytopathologist. Yes, that basically means he worked on plant diseases. But there's really no evidence that he had much of an expertise on thermal needs of trees or did any sort of validation or any experiments to develop this model. But there's something here that we can draw from the work of this guy, and that's this quote here, which I'm going to read to you. Although we may not understand why, when the plants differ from the expected reaction, we must remember that the plants are right and we are wrong. So moving this into the chill modeling space, it probably is an indication if the trees in Tunisia aren't doing what we expect based on the evidence we have, it probably means that we have to revisit the models that we're using and see if maybe we have to adjust our understanding. And so this has happened occasionally. There are a number of chill models. I know you have slightly different ones in South Africa that I'm not going to touch on, but the 3 major ones that have been used in the scientific literature are really the chilling hours model that I just briefly mentioned already. And there's the Utah model that you call a Richardson model, I believe, largely in South Africa, and then the so-called dynamic model, which to me sort of is the current state of the art, but it's actually not that current. It's from 1987. So all of this is pretty old stuff, and it's possibly not totally up to date when it comes to the science of dormancy. So this is how these models work. Yeah. So the chilling hours model, I already told you, a chilling hour is an hour between 0 and 7.2 degrees. This kind of looks like a computer science model with like some bit going from 0 to 1 or something. It's a very binary process. This is not really what biology does in most cases. And then here's the Utah model. You see there's sort of still kind of a pretty boxy structure, lots of right angles, which is also not a very biological idea. But we kind of prominently have here this negative influence. Yeah, so these kind of warm temperatures here can actually have a negative influence on chill modeling. That's sort of the new addition here that has actually caused some issues in South Africa. I think Nigel is going to mention that later. Then here's the dynamic model. Yeah? So this it's a bit more complicated to get this response curve out of the model. But here you see a smoother curve that sort of looks a bit more like what you could possibly imagine a biological process to look like. So these are the three ones, and just a few words on the dynamic model, because it's kind of my favorite. I'm going to tell you briefly how this works. Basically, it sort of relies on more complex assumptions. Basically, the idea here is that we first have some sort of a precursor of a chill portion that forms in the buds, and then by cold temperature, this then gets transitioned, gets converted into an intermediate chill product that can be destroyed again by warm temperatures. So to some extent, this intermediate can be destroyed completely. But once it's been converted to a chill portion, which happens if we have a certain amount of this intermediate, if it becomes a chill portion, it's fixed and basically builds up until the end of the season. There's pretty complicated equations behind this, and I sort of understand why it hasn't been taken up. But there are some tools now that make it easy to use. So if there was more interest in South Africa to apply this in practice, it wouldn't be so difficult to design tools that actually make it usable. What we found in quite a number of studies that the chill models aren't equivalent, and this is the main problem. So quite often when we try to model something, try to understand something, we look for some model and then assume, oh yeah, we found a model, let's use it. But it actually really matters which ones we use. The models are not at all interchangeable. And what we did here a while ago was to basically just calculate the ratio between two chill metrics. And what we're seeing here is the chilling hours, the number of chilling hours for about 5,000 weather stations around the world, divided by the chill portions that the dynamic model calculates. And you see there's a huge variation here in kind of the in this ratio. That basically means that here in Europe, maybe one chill portion corresponds to about 20 chilling hours. In South America, here this Chile was mainly the focus here. We have about 15 chilling hours per chill portion, and in some other places, including South Africa, we would have about 5 chilling hours per chill portion. Yeah? So basically, it means that it's sort of impossible that both of these models are biologically correct. They obviously seem to be expressing something different. And it also implies big problems when we transfer a cultivar from one place to another, and we have an estimate of the chilling requirement. It may not at all be exactly how we may not behave like we would expect it to behave in the place that we're taking it to. So the models we use really matter. But developing new models is pretty difficult because tree dormancy is actually quite complex. I mean, it remains relatively easily understandable when we just talk about chill and heat accumulation, but there's a lot of stuff going on in the plant under the hood. And I mean, science has focused a lot on these details. I don't even want to go into any details here, but you have, well, what happens basically is things like the transport of substances and information within the plant gets blocked to some extent by physical barriers. We have things going on the hormonal level. We have genetics and epigenetics coming in, carbohydrate storage. And there are a lot of things that seem to be interacting, and all of this sort of matters. And what modelers really like to do is make a model that really includes all the physical and biological processes and is actually based on an understanding of this. But it's sort of, it's a can of worms at this point, and we're not anywhere close at all to achieving this. So this is very complicated, and we really don't understand this well enough to model it. So for now, we're really stuck with this idea of the chilling and the heat, and I think we're going to remain there for quite some time. Now, of course, we don't have to understand all of this. And basically, what growers need is something that works and allows us to make predictions. And there, of course, we can make some progress. But it is important to remember that all these models are approximations. They're proxies. They're not actually reflecting much biological or physiological reality. And all the models have actually done okay in various places. I mean, the reason why these models are around even almost 100 years after they were developed is that they are working in certain places, and they allow us to make predictions that allow us to improve our management. But this is largely true under stationary climate conditions. Basically, if climate stays the same as it always has, then the experience from the past and kind of all the relationships between the models and reality that has been established in the past, that can guide us into the future if nothing changes in terms of climate. Now, the challenge is, however, that the climate is changing. Things are moving away from this stationary state, and we really need models now that remain useful in the future. So we need models that don't only tell us what the relationship is between reality and well, these models in the past. We need models that actually remain accurate as the temperatures increase. So models that are robust across climate gradients. And this is relatively difficult to develop. And of course, based on kind of historical observations, there's only in one particular place only, there's only so much we can do. And this is why I very much welcome the efforts currently in South Africa, coordinated by Esme and her people, to collect data and observations from different places across climate gradients. And if we get bloom data and temperature data from different places, warm places, cold places, which in South Africa you have lots of, then we can actually start making models that are reliable across these climate gradients and allow us to make adaptation and management decisions even in a warming climate. So if there's any way that you're not involved yet in this effort and any way you can contribute to this effort, I very much urge you to hopefully also on behalf of Esme to get in touch and see if you can contribute to this effort, because this is really, in my view, especially in a somewhat marginal chill area like South Africa, this could be really critical for adapting our orchards to the warming future. Now, there are some people out there who are even more critical, I believe, and have more insights about chill models, especially under South African conditions. And one of those is Nigel Cook, who's also here and is going to talk a bit more about details in the South African context now. I'll pass over to you.
Thanks, Eike. I'm live. Right.
You're live.
Okay.
Take it away.
Good. Yeah, thanks, Esme. Thanks, Eike. So, yeah, what is wrong with our chill units? And, you know, this is something that we've been struggling with for a long time in South Africa. And we all know just practically trying to draw information from one area's chill unit accumulation to the next one, always been a challenge in our situation. We are often in a situation by the middle of winter deciding what are we going to do with our rest breaking strategies, and you know we look at these things and we make the best guess we can, and sometimes it's not so accurate. So I'm just going to go through the details of where we've struggled. I'm going to repeat a few things. So I'm going to go back here, just in my first slide, to Jacques Raby's old work. He did this work in Jean Blou in Belgium. And what he did is he had apple trees growing outside, and he transferred them at different times during winter to a greenhouse, which was fairly rudimentary by greenhouse standards, but the temperatures were above the chilling temperatures. He managed to keep the temperatures around 15 degrees. So if you look here, if he transferred his apple trees that had stopped growing, they'd made buds, they were about to start losing leaves, and he put them in the greenhouse in September, and within 1 week, most of those plants had grown. Then next month in October, he put the same plants in the greenhouse, and they took 8 weeks to start growing, and they grew relatively poorly. Not all the plants started growing. And he did it again in November, December, January. And as the winter progressed, he noticed that the plants were growing better and better. And he came up with an argument that it was cold temperatures causing the plants to go dormant, and cold temperatures that, again, brought them out of dormancy. And he backed this up with a very simple experiment. He took some of these trees in September and put them in a fridge at 8 degrees, and 2 weeks in the fridge and 1 month in the fridge made the plants go more dormant. So he argued at the beginning of winter, chilling temperatures are making the buds dormant. And then he took plants that had been outside until November and he gave them the same treatment. And after 35, 45, 60 days at 8 degrees, they were able to grow faster. And this points to the fact that chilling temperatures, what we refer to as chilling temperatures, can make buds go dormant and bring those buds out of dormancy. Okay, but it didn't take very long for things to get complicated. And this is work that was done after that by French researchers, Morget and his colleagues, and they were working on walnut plants. And they did the same kind of experiment. So they put plants from outside into a greenhouse, again, warmer than 15 degrees. And every month they put plants in there, and they could see that in the beginning of the winter, the plants went dormant. And then somewhere in December, they'd reached a point where the plants started to come out of dormancy. And by the time that it was spring, the plants were able to grow well and fast. Bud burst was rapid. What they also noted is that if you put plants in a warm environment in this greenhouse and forced them, and they never received any chilling, that they still went dormant. Okay? But the process to go dormant took longer, and then the process to come out of dormancy at these non-chilling temperatures also took longer. But the plants still went dormant, okay, at non-chilling temperatures. So what the chilling temperatures seem to do is they seem to make the process go faster. Ever since then, we've known that there's the central role for temperature involved in the process, and we've struggled with it, as Akers pointed out. This is some South African data that we collected years back and published, and I'm going to go through this because I'm going to come back to it later. What we've got on the graphs is these first two graphs are Golden Delicious. These are Golden Delicious, and these two graphs are Granny Smith. And the top two graphs are plants grown in Elgin, which we've called warm, and then in the Kobokkeveld, which we called cold. So the dots on the graph are Utah model Richardson chill units, and I'm only really going to present data with the Utah model. With its shortcomings, I'm just going to stick to Utah just to make my part of the conversation a little easier. What we see with the Golden Delicious in the Kobokkeveld is at the beginning of autumn, the buds grow fast. You can see these two dots here. The plants were able to grow within 12, 15 days. And then all of a sudden, a week later, the plants are dormant. And then after that, the plants slowly exit dormancy as they receive chilling. And then at the end, they exit dormancy a bit faster. But at the end of winter, the plants have exited dormancy. They're able to grow normally and rapidly as the chilling accumulates. And you can see in series that year, we got to about 1,500 Utah model chill units. Okay, let's look at what happened in Grabo. The same thing happened, the plants went dormant. But they didn't go dormant as quickly, and they continued to go dormant through winter. And they only reached this maximum depth of dormancy, or their deepest winter rest, by July, which is pretty much more than halfway through winter. And in fact, the plants have already accumulated 400 chill units, roughly, or 500. And then the winter's obviously not as cold, it's warmer in Elgin. They start to come out of dormancy, and this area got to 800 chill units that year, and the plants were still not completely out of dormancy. In human terms, that we'd call them half asleep. They take around 20, 25 days to grow, as opposed to less than 10 days in series. These plants are still in some level of dormancy. Okay, I'm going to come back to this one. I need to talk about chill units. Thanks, Aker and Esme, you've done a lot of it. The Utah model, I've drawn a curve. You're absolutely correct, Aker. It is a stepped model. But what Utah says is every hour at 7 degrees is equivalent to 1 chill unit. Every hour at 14 gives you no chill units, and every hour at 21 degrees gives you -1 chill units. And what you have to do is add up all these hourly units for the day, all 24 of them. And if temperatures are warm, if you're at this end of the spectrum, you're going to end up with a negative figure. And if you're at this end of the spectrum, most of the day, you're going to end up with a positive figure. There was research done that said we need to go to 28 degrees and -2, but that's not been incorporated into the Richardson model. We have a strong suspicion in South Africa that when we do hit these kind of temperatures, which does happen in areas like the Klein Karoo and Wellington, we get these 28-degree days in July and have a nice spell. And it seems to be far more detrimental, which is what the potential adaptation to the model suggested. This piece of the graph is not included in Utah, and it's not included in my data. Okay, so we're talking about this curve from 0 to 21 degrees. And most of the models work on a similar curve and add up chill units in different ways. I'm not going to get into that. I'm going to stick with Utah. Right, so let's get to the meat of it. What's wrong with our chilling units? Man, I'm going to refer to the first problem as an honest mistake, and I think sometimes in the past we've just got the data wrong. This was last year's data from May until July, and you can see that we're at around 240 something for Elgin, 230 for Greyton, Villiersdorp 290 to 300, and then if you look at Fairboom, we're at 650. Okay. Now, last year in the middle of winter, the guys in Fairboom were telling me they're going to cut back on their rest breaking. They're having a really cold winter, but it doesn't make sense because the climate doesn't differ that dramatically from Fairboom to Villiersdorp or to Elgin. And the next slide points out where the error is, and like I say, this is an honest mistake. The average temperatures didn't differ, and something had gone wrong with the calculation and led to some quite difficult conversations with growers who think that they've had 600 something chill units and are now going to spray less rest breaking agents. So yeah, mistakes happen. We're not going to hold that against anybody, but it's something we need to be careful of and something that we need to do our best to prevent. Right, let's carry on. This is Elgin Orchards weather station. The next mistake that happens, if you go to the original paper published by Richardson, then what they tell you is you wait for the point in the year where the model goes from accumulating a negative figure on a day-to-day basis to the point where it accumulates a positive figure. And that is your day 0. That's, according to the model, the start of the chilling period, and you add up from there. So I just want to show you in this purple year, which was 2019, I think, we accumulated some chill units early in April. And we accumulated, we dropped, we accumulated, we dropped, and then we started accumulating much later. And that's very typical for the Elgin climate in autumn. We can have a cold front, it gets properly cold, and then we have a week of nice weather and it warms up. But in 2019, we started in April. If you look at the 2020 curve, the black one, the first time we received positive chilling units was at the end of May. And there's a big difference in the start dates, and that needs to be respected in the way that we record chill units. And I'm just going to show you some other data. This is again industry accessible chill units. You can see in the 2015 year, I think it is, 2015, doesn't matter. Maybe it's 2019. The chill units only really started accumulating at the end of May. The problem is, if you start on the 1st of May, which we have done historically, you're going to accumulate a negative 140, which is going to come off your sum. So you have to start each weather station at its turning point, and you can't just start accumulating chill units at the 1st of May. In fact, what this means is this is not actually Utah model, it's not actually Richardson units, it's something else. And our way of reporting chill units is not actually, it's not actually recognized by any of the models. The downside of this is that every weather station has to be calibrated for every year, and you need to calibrate the start date. If you look at the variation that I've noted in this era, this could lead to somewhere in the region of around 200 chill units less than what we've actually accumulated, comparing years on years. And yeah, we need to change this part of it. Right. The next part of the problem, this is the work we did with Laura and Esme, where we collected shoots from all over the country, and we said that the buds are behaving different in the different areas. And I'm going to summarize it quite simply. We've got the Free State cluster of dormancy curves, which start earlier and go into a deeper dormancy and then come out quicker. Okay, and this is averaged over 5 years. And then we've got the blue line, which is the series. They start a little bit later. They go, the green line is series. That would be series Coe-Bockerfeld. They start a little bit later than the Free State, but they go into dormancy and they come out not as quick as the Free State, but definitely quicker than the Hortgro area. And we see that when the climate is warmer, that the entrance into dormancy is later and the exit is slower. We've suspected this for a long time, but what we also see in nearly all of these scenarios is that the plants are dormant pretty much before we've accumulated any significant chilling. Okay. There's a whole lot of physiology and morphology and plant life happening in the process before it gets cold enough to accumulate chill units. Okay, I'm going to come back to this graph, and I've already alluded to it. In series this year with the Goldens, the buds were properly dormant before any accumulation of chilling, whereas the same variety in Hortgro accumulated 400 chilling hours before it was properly dormant. Okay, and it's quite simply put, Esme's done some recent work, which she's about to publish, just to put you under pressure. What we know is that the Utah model chill units and Infratex the same, it doesn't work really in this first part of the dormancy curve. In fact, Esme's work has shown us that there's a very, very poor correlation between the accumulation of chill units and the entrance into dormancy. But from maximum dormancy coming out, we know that the chill units do work, and that they are reasonably accurate in the second half of the curve. The problem is that we don't have any morphological indicators to give us this date here. And it would appear that we need a different model to determine when this point is, so that we could then start using chill units to predict the exit from dormancy. So I realize I'm not offering many solutions. Aike is going to calculate a new model for us, and hopefully the new model will solve all our problems and help us with our rest-breaking predictions, which currently is a little bit of a dark art. Yeah, so that's it from my side. Yeah, thanks, Esme.
Thank you very much, both of you, for trying to unravel what we're dealing with. And whilst I was listening to you, I realized again that something seemingly simple has got very complex biology that we cannot see and that we cannot seem to find a handle. We've got a plant that needs a lot of chill to perhaps go into dormancy, or maybe it does so just genetically, but it certainly needs a lot of chill to come out of dormancy and be released from inhibition. We know that this differs from one species to the next, the chill requirement of that specific species. We are also able to measure the environment and know what the chill is on offer in a certain area, and we try and match those two up. But yet we don't quite understand. There seems to be more questions than answers. What definitely comes out of this is the tools that we are using, these chill models that we are using to try and describe how the plant interprets, accumulates, banks, collects, remembers, all of those things, the cold, the chill that happens. The tool that we are using doesn't seem to be accurate, especially if we work in warm winter areas. It's the best tool in the shed, but it doesn't seem to be very accurate. And also we see that thinking forward into the future, having even warmer climate, with climate change happening, this problem is not going to go away. It's actually going to become worse. And I think in South Africa, it's been happening in our backyard for a while, but I think Northern Hemisphere countries are very quick in realizing that this is coming to their orchards and coming to them pretty soon, and therefore this renewed interest in dormancy. And again, the search for a better tool. The search to find something that we can handle, something that we can use to quantify this reaction. And we currently have a project that we've launched about a year or so ago that we would like to investigate exactly this. We've seen that there are many invisible things. We see that there's difference in temperature. We actually want to go one step back and try and simplify by going back to look at what the plant tells us in terms of bud break. We call this the phenophase temperature database that we want to develop in South Africa for the South African industry. If you could just pause it. Thanks. Phenophase temperature database. This is a Hortgro sponsored project between the University of Stellenbosch and then also Provar. Myself and Dr. Ivan Labuskachny are the project leaders on this phenophase project. And what we are trying to do is we've established sites. We've established trial sites for apples and plums. And we've planted 10 different cultivars. That's up on the screen now. You can see a range. For apples, this ranged between low chill, medium chill, and high chill cultivars. For plums, early, medium, and late flowering plums. We planted 10 cultivars across these ranges, and we planted them in 3 different areas. We did this a couple of years ago. The plants are currently going into their 3rd leaf, so there you can see the progression to where we are now. And the plan is this. The plan is to monitor these trees, all these different cultivars, in 3 different areas. There you can see the areas Nooit Gedacht, Lisshof, there's the Boerland Landbou site, there's the Oak Valley site, and then there's the Klibos Laagte site out towards Robertson. This project also includes cherries, that's why you will see the cherries up there. But we've planted these 10 in a randomized block designed to satisfy all the statistical requirements in all of these areas. In other words, we've got cultivars in areas where they are well adapted to, and we know that they will do well, but we also have cultivars in areas where they would definitely not grow well. They would definitely not cope and are definitely not adapted for that area. And that's exactly what we want to see. We want to monitor the bud break. We want to see how these plants respond in terms of their bud break. This trial also will contain a split plot, so we will have some trees being treated with rest-breaking agent and others just without rest-breaking agent, just the temperature as it is. And we want to try and understand what happens, and we want to then calculate the chill units, all the different models that we could possibly do and that is known to humankind by now, and compare that, link that up to the bud break that we see in these areas, link it up and see if there's a correlation. To try and understand the relationship between our models and what the plant is actually doing when bud break happens. And then from that, try and determine which of these chill models are the best for these specific areas. That's the one aim. The other aim is also to perhaps use the data that we're generating long term and try and develop a model that will also look at chill units as well as heat production in all of the different seasons and link that again to bud break. So we're coming back to basics by coming back to what the plant actually shows us during bud break, how it responds to the winter, the autumn and the winter prior to bud break. So that's what we're trying to do. I hope that this will help us in collaboration with ACRE and input from industry to try and understand what we are dealing with here, and hopefully in the future try and put together a better suited model that is based on actual bud break phenology data and not just temperature data or numbers, but rather more what the plant is telling us directly. I want to, now for the important part, I want to open up the floor for questions to the speakers. So maybe you want to prepare that. That last slide that I had up a while ago just explains that everything we've been saying here today actually appears in the Fresh Quarterly of June, which is out now, I believe, Hortgro Fresh Quarterly. There are 4 articles on dormancy. And just get back to that slide for a moment. Show you one more slide, please. Let me move up one more slide. If you download the Fresh Quarterly, you'll see that there are 4 different articles on dormancy, and it basically follows the same narrative that we had here this morning. Started off by giving you the basic understanding, then moving over to ACRE's section on chill models, and then Nigel's section on what is wrong with chill models. There we have it, up the Fresh Quarterly. So these 4 articles were written by Anna Besson after she interviewed all of us, and we put these articles together. The 4th article basically ends with the phenophase project that I've just been talking about, and how we are going to try and address the challenges that comes with warm winter areas. So now the floor is open for questions. I see we already have one question from Jacques. I'm trying to read on the screen here. Are there no other phenological markers that can be used to track dormancy, like starch accumulation, bud size, or others? Jacques, thank you for the question. Maybe I can answer that one. Yes, I'm sure there are markers that one can use, and definitely been seeing that. I'm just thinking off the cuff of a specific protein, a 91 kilodalton protein that's been identified that seems to be increasing and decreasing when we look at dormancy. We've certainly done work on looking at changes in respiration rate, changes in lipids and membrane composition and makeup of that. So yes, there are. The problem, however, is that these methods are always retrospectively. There's no immediate marker where you can walk into the orchard and identify and say, this is where the orchard is right now in terms of that dormancy cycle that it's going through. It always appears to be retrospectively, in other words, we collect the buds, now we've got to go and do the tests, and then maybe weeks later we get an answer to what the dormancy status is, and then it's very difficult to still have an immediate reaction. So yes, there are markers, but they are not real time, and I think that can complicate giving us direction for that very season that we are in. Don't know, Nigel, if you or Aker wants to add to that. I see there's another question. What should the producer do in the absence of perfect chill models? So what are we to do? Where does that leave us? How do a producer with an orchard watches these reports coming through, realizing there's not enough chill? What do I do? How do I rectify that? Nigel, I'm looking at you for that specific one. You want to tackle that?
Yeah, I think that the way to do it is to make sure that you are, first of all, recording the chill units correctly. That's the most important thing. The second thing, and I do this all the time, is I don't look at a weather station in series and try and make a conclusion about what to do in Wellington on plums. I think if you have a local weather station or a weather station on the farm, then it makes a lot of sense to compare what's happening on that farm over years. So like the graph I presented with the Elgin Orchards data, we record that on Elgin Orchards for Elgin Orchards in real time. And by the time we get to the middle of July, end of July, when we need to take decisions on rest breaking, then we're making decisions for the farm based on the farm's track record. So it's not 100% accurate, but it does tell you whether you are generally worse off or better off than last year. And you get a rough idea for whether you should be stepping on the gas or stepping on the brake.
So there's no one size fits all. It's a very unique kind of management, precise farming, precision farming.
Yeah, I think the worst mistakes come in when you start comparing, comparing Elgin to series. That's where it starts to get shaky.
Mm-hmm.
I see another
just one addition to this. I think it is possibly important to have multiple chill metrics in your view as you kind of accumulate experience over time. Maybe not every grower, but I think we should be researchers maybe who monitor how they perform and collect experiences maybe from practitioners. I mean, if you feel like it's not working well, then somebody needs to know this and use this to validate and possibly adjust the models we use. I think it's important to collect experiences on what works and what doesn't, because the models aren't perfect, but some are better than others, and some may be better suited in some places than others, and it's important to collect this and experience. Let us know.
I think that's exactly what we're trying to do with this phenophase project, is actually taking that route, looking at all the models and looking at all the different areas and all the different cultivars to actually pinpoint which models are best suited for a certain cultivar or area, etc. I'm going to move on to the next question, thanks. It says, is KwaZulu-Natal inland cold places like Pietermaritzburg good for starting an apple farm? You can see it looks like we've got entrepreneurs wanting to start apple farming up in KwaZulu-Natal. What do you advise, Nigel?
Yeah, I think we've got quite a lot of experience in Natal and apples. It's not a mainstream apple production area, but there's been a fairly long history of trialing apples. I myself have been involved in apples in the Camberg, in the Middle Risk area, in the Franklin-Swartberg area, and in the Underberg area. And the apples do quite well there. I'd say Pietermaritzburg is probably getting a little bit too warm for the mainstream apple varieties, but the newer low-chill apple varieties would work well on a good site in Pietermaritzburg. Yeah.
Thank you. Another question?
Maybe it's for me.
From a modeling perspective, I don't know if you can see that question, Aker, from a modeling perspective, this is really opening exciting doors given the climate database for the present and the future, which we have access to. at fine spatial scales. Do you want to expand on that, perhaps?
I can answer that. I think that that's absolutely true. I mean, we've done, I didn't show that here, but we've done some mapping, and we had like a global map of kind of future chilling. That's certainly true. I mean, the challenge, of course, with these climate data sets is always that they have to sort of reflect the reality on the ground. They have to be reasonably accurate, and they have to be at the right resolution. But there certainly is a lot we can do. The challenge to me primarily, and this comes back to what we've been talking about, I think we have better future climate data now than we have chill models. So there's always sources of inaccuracy, and we always talk about climate scenarios and all that stuff. And yes, there's uncertainty there. But I think there's possibly more uncertainty that comes in the process of converting temperature projections to chill on the ground.
Thanks for that question. I see that it's from Professor Schulze. It's also involved in modeling of temperature. Thank you very much. I think there's room for collaboration there as well.
I was happy to. Mm-hmm.
Another question, maybe the one on screen this side. Is it a correct deduction that autumn temperatures are really important? Are autumn temperatures, we know that winter temperatures are important. Are autumn temperatures important? Maybe I can start off the conversation. We are currently, or we've just completed a 2-year study where we actually look at autumn temperatures versus winter temperatures versus spring conditions or heat accumulation in spring. And I'm actually jumping the gun here, but what the data seems to be showing us is that if we have a cold enough winter, and under such conditions, the shoulder temperatures, shoulder seasons are less important. The autumn and the spring seems to be less important. The winter is the overriding factor. But as we move into marginal areas with warmer winters, these shoulder seasons do become important. That much we've seen, that's when it can play a role, where how quickly one starts accumulating chill in autumn can contribute, as well as the amount of heat units we again see in spring. So that becomes important there. Nigel, have you perhaps seen that in the field?
Yeah, no, I think at the end of the day, the winter temperatures are definitely the most important. And I'd say on the lines that, you can have a good autumn, but if you have a bad winter, you're going to have a bigger problem than if you had a bad autumn and a good winter. Obviously, the autumn and spring temperatures also play a role. Yeah.
I think that is also something perhaps when we do our modeling, that we should start looking at that in more detail. What is the contribution of the temperatures that we experience during autumn versus that of winter, and also in combination with? So it calls for a much bigger analysis of temperature, not just a fixed number, but maybe look at how the different seasons contribute and how that works. There seems to be quite a lot of questions coming through. This is great. Can the industry graphs include both the Utah and the dynamic model in the future? So there's a question out to Hortgro that's providing this information, and I can see Vian off screen nodding his head. I think if anything, this should be the starting point of today's conversation, or the take-home message, that we should definitely be open to look at more chill models and not just the Utah Richardson chill models. I think Ake has illustrated how these models evolved, and that there are better tools or new tools in our shed that we can use. They're not perfect, but and they could also be custom for a certain area or certain cultivar. So I don't think it's a one-size-fits-all. Ake, you want to?
Yeah, sure. I can. I mean, so there are also questions about the dynamic model that, of course, are still haven't been answered. I mean, this was developed in Israel for peaches. Yeah? So most trees that we're interested in aren't peaches, and they're not in Israel. So it's certainly, and there are some parameters in this model that we should be adjusting for each cultivar and each species. We're actually about to publish a paper on a framework that can possibly achieve this, but then it raises all kinds of other questions. Maybe we'll need different chill units for each cultivar, and that sort of then becomes a huge mess. But I think there is a lot of potential and a lot of things to explore, and I hope our new framework will help out a bit, maybe in combination with the data sets that you guys are collecting there. I think there's a lot of scope for breaking new ground pretty soon, hopefully.
And then our next question I see is from Chad, MEC student of mine. Welcome, Chad. Good to see you in the conversation. How accurate is our understanding of the heat requirements of the fruit we grow in the Western Cape? Will the Phenophase project aim to provide more info on the heat aspect? I think that's a very important question. I think the models are currently very focused on chill and the accumulation of chill. And I think to a degree, maybe some of the more modern, and Ake, you can correct me here, there are some more modern models that are now also starting to incorporate spring temperatures or heat units more directly. I certainly think there's more room for that. And linking that up to bud break, as I've been saying, would definitely shed more light on our understanding.
Yeah, I think heat models is what we haven't really looked at very much, and I suspect there's also a lot to discover there. I mean, I'm not entirely sure on where exactly the models come from that we're using, but I suspect that they're the same models that we use for modeling a growing tree with leaves and photosynthesis and all those things. And I'm not so sure that they respond the same when they don't have leaves as they do during the growing season. But it hasn't really been the focus. So far, we're assuming that also here that every tree we're using is a peach from Utah, I think in this case. I think this is where the common models come from. Also not.
Yeah, just on the heat unit thing, and just from a genetic perspective, we know that there's as much variation in chilling requirement as there is in heat requirement. And we also know that varieties don't inherit per se a low chilling requirement with a high heat requirement. The inheritance of the two traits is very poorly linked. So it becomes very complex. And I think this is definitely, I didn't want to get into this level of discussion on chill units, but going forward, the heat requirement needs to be acknowledged as much as the chilling requirement.
Definitely so. Oh, I've got questions coming at me from all sides. This is absolutely great. Let's take the next one from Peter. Do varieties have a fixed chill requirement? And that's an interesting one. Nigel, I think you can talk from experience.
I absolutely believe that they do. If they didn't, then a variety like Red Delicious, Top Red, would have changed in the Elgin area to become a medium or low chill variety, which it hasn't. Obviously, there are ways that plants can change chilling requirements, you know, through mutating, but those are slow, long-term changes, and in a practical sense, it means a change in the variety. So a variety, in my view, does not change to its chilling requirement. There are 100-year-old Enifefe Golden Delicious orchards in Hortgro, which have the same level of chilling problems as 10-year-old Enifefe Golden Delicious orchards.
This is actually quite difficult to evaluate, though, because there are, of course, studies from different places that indicate different chilling requirements for different cultivars. But now again, we have this problem of how we're measuring this.
Exactly.
And here again, we have a challenge of the models. I mean, if the models are useless, then how do we even start comparing between sites?
I agree. I also think that the chilling requirement is definitely set, but our interpretation or our measurement of that is very crude at this point in time, and our yardstick might not be the right one that we're using.
Maybe it's important to just say that I think the conversation around a chilling requirement is a very different conversation to the one that's required about a chill unit.
You're absolutely right. I see a message, or actually just a comment coming through that indicates that iLeaf, those of you that are using that software, iLeaf displays the dynamic model graphs under chilling portions icon. The Richardson and Infratech graphs is displayed under cold units for those of you. So that's just a comment. If you are using iLeaf, you can find the information there. what is this? Apart from heat, what might be the main improvements to the current models? So the models that we have, apart from heat, how else can we improve them? Eike, this one is coming straight at you.
Sorry, I was just distracted for a second.
So it's asking us, what else can we improve? How can we improve the models that we currently have? We see that they are relatively broken. How can we improve them apart from including the heat units? What else can we do to make them better, suitable?
Yeah. I mean, I think, so I always like, I mean, of course, we can have all kinds of controlled trials and really fine-grained temperature treatments and stuff like that. But I mean, I'm a friend of exploiting variation that exists. What I think we really need is larger data sets of temperature combined with phenology, and there's probably a lot we can draw out of this. If we have such data sets from different locations, different climates, I think that would be super useful. Of course, then there's the unfortunate South African practice of using rest-breaking chemicals, which makes some of the data less useful than it would otherwise be. But I guess it's not your objective to feed scientists, but rather to feed people. So that's okay. Keep using these. But I think there would be a lot. There will be so much value in the data you're currently collecting, and maybe it'll be possible to bolster this with additional observations from other places.
Thank you. Vian, I'm looking at you for a moment. I see that we are over our time. Are you okay for us to continue? Yes, we're getting the thumbs up, so let's try and look at some more questions. In the trials, would it be valuable to have a look at when the trees come out of dormancy, where their reserve status and even soil temperature is, and how this influences the results you get from what is measured to find out if you had good dormancy? Surely that also differs from site to site. So yes, I agree there that when we do this phenophase project and as we're doing it, I think one should try to measure as many things as possible. It looks like there might be other indicators that we are currently missing in the models, and whether that is reserve status or soil temperature or whatever the case might be. Some of these things are not always simple to measure. But I think the obvious ones we should certainly do, and also include even ones that perhaps don't seem to have a clear direct link to dormancy. Who knows, maybe those could also be indicators. So I think we would like to include as many parameters as we possibly can for that one. Next one, how will a model take the effect of fixed nets into account? This is an interesting one, a practical question. We are moving towards fixed nets more in industry for other reasons. Will that in any way affect the way we measure and the plant response?
Absolutely. So the nets change the environment. I'm actually going to try and answer the last two questions together there. Chilling models and chill units are calculated on air temperature, not on bud temperature. Okay, and the air temperature needs to be measured in a standard way, and that means that it's a certain height above the ground and it's in a Stevenson screen. So if you're measuring bud temperatures, then bud temperature cannot be used to calculate chill units. You're measuring something completely different. We do know that certain environmental conditions change the bud temperature relative to the air temperature. Under dry, sunny conditions, the buds are usually warmer than air temperature. On cool, wet, misty conditions, the buds are usually cooler than air temperature. And this has been long reported to have been one of the sources of variation between chill units between areas. But it's very difficult to have a very simple model and take all of these things into account. Yeah, there are no models based on bud temperature. I don't know of a single model that's based on bud temperature. I know that people measure bud temperatures, and they do that with overhead cooling, and then they say that they've increased the chilling. You do, because the buds are colder. But you'd have to use a different set of parameters. You know, so I think these are the kind of fairly obvious limitations of chill unit models.
So again, points towards the inaccuracy and
the other one that's just as common is in most warm climates around the world, we use rest breaking agents, not always Dormex, but we use rest breaking agents. We use defoliation, we use pruning, we use nitrogen containing compounds and other rest breaking compounds. So what we are doing in the warm areas is we are manipulating bud burst. And often you will see climate data reported to have 800 chilling hours, and the guys farming a variety with 200 chilling hour requirement, and you know that bud burst on that variety was at the end of July, and the data that's presented was recorded until the end of August. Obviously, it's meaningless. You know, once the plant starts growing, there's no point in continuing to measure chill units. I go so far as to say once you spray rest-breaking chemicals or defoliate or start manipulating in any other way, that you might as well stop recording the chill units and rather look at heat units and other factors that promote growth.
I agree. I think one of the take-home messages also from today is that, yes, the chill models we have are not the best. They're not perfect in any way, but it also seems that the way we apply them, these broken tools, the way we apply them is compounding to this, is making the problem worse, because we're not using, although they are already not accurate, the way we use them is making it even worse, because we are measuring temperatures that we shouldn't be measuring, or dates or time spans that we shouldn't be measuring. It seems that maybe for starters, for lack of having a better model, we should pay attention to which models we use and make sure that the ones that we do use, we use them correctly. So I think for the time being, that's probably what we need to make sure that we do that correctly. And hopefully we will be able to generate a better understanding of these models, and hopefully build new models to take us into the future, because we know with climate change that this is really going to just get worse, and we need to prepare ourselves for that. I want to thank everybody, especially the two guests, Eike and Nigel, for sharing knowledge and helping us unravel this, and everyone for the very insightful questions. And it's just an exciting topic that unfortunately has more questions than answers, seemingly simple, but really complex. Thanks everyone for joining in. Appreciate this, and we will continue to serve through research. Thank you.
Thanks, Esme. Thanks, Eike. Thanks, Hortgro. This was, I think, an important conversation, because every year there's confusion around this subject, and because of shortcomings in the models, but also because of the way we go about doing things. So from my side, thanks. I really appreciate it.
Well, thanks from me too. It was a pleasure talking to you guys, and all the best with well, the upcoming spring and dormancy completion and all those things. I guess you're just getting started now. No, you're really in the middle. You're getting in the middle of the dormancy, right?
Yeah, we're
convert
towards winter, the other way around.
Convert dormancy. So all the best with the upcoming dormancy. I hope you'll be measuring and recording all kinds of things and sending it on to the scientists.
Thanks, Eike. We'll be in touch soon.
All right. Bye-bye.
Thank you, everyone. Thanks for participating. Goodbye.